proteins are often described in levels of structure, sort of like how I could describe you on different levels. I could describe you on the chemical level and say that you're made out of hydrogen and oxygen and nitrogen and proteins and carbohydrates. I could describe you on the cellular level if I took a step back. I could describe you, on the organism level, on the tissue level. And so proteins are described by these levels of structure as well. So the first level of structure in proteins is called the primary level, and it's represented with a 1 with a little degree sign. That means primary. And the primary level, every protein has one, is the order of amino acids in that protein. And that's actually going to determine the ultimate structure and function of the protein. The order of the amino acids, the primary level of structure, is the most important level because it determines all the other levels. So I have a little section of protein here. So imagine this was a little protein chain, and the primary structure of this protein chain would be basically the fact that it's made of 4 amino acids, and then what specific 4 amino acids those were. So here's our first amino acid, and then a peptide bond, hooking it to the second amino acid, and then the third amino acid, and then the last amino acid. And these are the specific R groups. So this is a specific amino acid in position number 1. This is a second specific amino acid in position 2, a third one in position number 3, and then the last one in position number 4. And again, if you had to count how many amino acids were in a chain, it's obviously pointed out for you here because there's blue boxes around them. But if you just saw a long chain, if you look for that repeating pattern of NCC, because remember, every amino acid is going to start off with an amino group, a carbon in the middle, with an R group, which might actually be drawn out like it is in this picture, and then on the other end, a carboxylic acid group or a carboxyl group. So even when they hook together, you're never going to lose this backbone. It's always going to be a nitrogen, a carbon, hooked peptide bond to the next one, nitrogen, carbon, carbon. And you can see it here, nitrogen, carbon, carbon, peptide bond, nitrogen, carbon, carbon, peptide bond, etc. So this is the primary order, and this is determined by your DNA. Your DNA, remember, is a code that tells your body you're going to make insulin. Insulin is going to have these specific amino acids in this specific order. Remember, there's a total of 20 different amino acids that are possible, but proteins are typically 500 or more amino acids long. So you're going to reuse some of those 20 amino acids in a chain. You can see the same amino acid several times in a row, but it's all going to be very, very specific depending on what protein it is. So that's called our primary structure, just going all the way down to the molecular level and just listing the order of amino acids. Now, once you have this chain of amino acids, it starts to fold up. And the first thing that's going to happen is the amino groups and carboxylic acid groups are going to react with one another. So remember, every amino acid has an amino group on one side, and then our R group and our hydrogen, and then a carboxylic acid group on the other side. And here's where this amino acid is now connected to other amino acids in the chain. But this hydrogen in the amino group is polar. It's very interested in making hydrogen bonds with oxygen. And this oxygen is also polar. It's interested in making hydrogen bonds too. Now, it can't make a hydrogen bond here because these are too close to one another. But what does happen is amino groups and carboxyl groups further down the chain react with each other. And there's 2 specific shapes that we get. One is called the alpha helix. Notice that little alpha symbol we talked about yesterday. So this is the alpha helix. Kind of looks like a curlicue, like this. And again, notice, see, hydrogen bonds between hydrogen and oxygen. This has nothing to do with R groups. This is just amino and carboxylic acid groups along the chain. Another possible pattern that we might get is called a pleated sheet, or sometimes it's called a beta pleated sheet. And this one looks kind of like an accordion, where it kind of folds back and forth, back and forth, back and forth. But again, it's caused by hydrogen bonds between the hydrogens of amino groups and the oxygens of carboxyl groups. And this is only in sections of a chain. In fact, let me show you the next slide. It kind of shows you what I mean. So the primary structure was the order of amino acids. Here's some secondary structures here. That would be an alpha helix, just in one little section of this chain. And these are all pleated sheets in sections of a chain. So the secondary structures describe how portions of a protein chain fold up, not how the whole thing folds up. Our next level of structure is called tertiary, and that's actually what this slide is showing. So tertiary structure is how the entire protein folds into its final shape. One chain folds into its final shape, and that is based on specifically R groups. So this is where the R groups become super, super important. So this picture at the bottom shows some of the possible interactions that we see between R groups. One of the most important ones is the sulfhydryl or thiol R group we talked about. It was one of the R groups you had to remember as a functional group. And if you look down here, it actually forms a covalent bond called a sulfide bridge or a disulfide bridge. This is a covalent bond. This actually is one of the most important bonds in stabilizing a protein into its folded shape. But in addition to that, we also have hydrogen bonds. Here's a hydrogen bond between two R groups this time. This is a hydrophobic interaction, meaning this methyl group, or the methyl groups here, are hydrophobic. They don't like water. So what they're going to do is they're going to be attracted to other hydrophobic R groups, and so this is going to fold into a shape. Down here is an ionic bond. So this protein is going to fold up into its final shape, not only because of the R groups interacting with each other, but the fact that these R groups don't like water. And you remember, you're 70% water. So when this protein folds up into its final shape that looks kind of globular, but it really is a very specific shape, what's facing outward is going to be all the polar R groups, because they like water, and your cells are full of water. And in the middle of this protein are going to be all the hydrophobic ones that don't like water. And this is why it's so important if you were to have a mutation in your DNA, and let's just say right here in this spot, the wrong amino acid got put there. And instead of it being a hydrophobic R group, a hydrophilic R group got put there instead. This protein would no longer fold here. It would now twist a completely different way because these R groups would no longer be attracted to each other. So the final shape of a protein, it's super important that the right amino acids are in the right spot. It all goes back to that primary structure. The amino acid, the order of amino acids in the code is very important. It's going to determine the tertiary structure. How does this protein fold up into its final shape? Also, tertiary and secondary structures are very susceptible to changes in temperature and pH. Specifically, a really good example of this is high temperatures. If you have a really high fever, like 104, 105, what's going to happen is a lot of these, especially the hydrogen bonded R groups, they're going to stop interacting with each other. So this protein that right now is folded into a very specific shape, it's going to unfold. It's not going to break the peptide bonds. The primary structure would not be affected. This would really affect secondary and tertiary structures where the hydrogen bonds that are very weak might not react with one another, and so this unfolds. If this was something that, whatever the job of this protein is, it can only do the job when it's folded into the right shape. So if proteins, for example, in your brain start to unfold, your brain is not going to function. You're going to have brain damage. And so high fevers can be very dangerous for all of the proteins, particularly enzymes in your body that are speeding up reactions, because if they get denatured, which is the word for when they unfold, then they're not going to be able to do their job, and you're going to suffer the consequences of that. pH changes will also change the activity. Sometimes you do that on purpose. Your stomach has a very low pH. That's on purpose because that low pH activates pepsin, which is an enzyme that digests protein. That enzyme actually only folds into the correct active shape when the pH is low. If you raise the pH, it shuts that enzyme down. So you can control the activity of certain proteins just by changing temperature or pH where that enzyme is located, or hormone, or whatever that protein might be. Finally, there's a level called quaternary structure. And quaternary structure, not every protein has this, but some proteins in your body are a little more complicated, that the final active protein actually consists of multiple chains bonded together. And the best example of this is hemoglobin. If you look at the picture of hemoglobin here, it's actually 4 chains. So if I just look at one of these chains, I'm looking at, this is the tertiary structure of this one chain, and this is the tertiary structure of this other chain, and then there's 2 other ones. But this hemoglobin, in order for it to function, 4 chains have to interact and form a final shape, and that's called quaternary, 4 with a little degree, quaternary structure. So those are the levels of structure of protein. I do have a little summary here. So the primary structure, that's the order of amino acids in the chain. So this chain may consist of 350 amino acids, and this is the exact order. It's lysine, lysine, glycine, glycine, leucine, leucine, alanine, histidine, blah, blah, blah. And if one of those was to change, it might change how the entire thing folds up later. Secondary structures are only found in portions of a chain. They're regular repeating patterns that occur, and they're showing the secondary structure here, how it's in just a little section of this protein. Tertiary is how the whole chain folds up, one full chain, and this one especially is due to the R groups interacting with each other. And then finally, some proteins have a quaternary structure, where the final functional protein consists of multiple chains all interacting. So those are the levels of protein structure, and you need to know each of them, what they look like, what causes them, and also know about denaturing, how that can change the structure.